All right, let's just jump right in. I want you to think of your DNA, your chromosomes, as this incredible architectural blueprint for life. Seriously, it's like a massive encyclopedia with every single instruction for building and running you. But what happens when that blueprint isn't exactly perfect?
And that's really the core question we're going to dig into. We're talking about all the massive, sometimes surprising and, sometimes revolutionary ways our genetic instructions can get altered. We're talking everything from a single page being moved to entire volumes being added or just lost.
Okay, so first up, let's look at the really big picture changes. And I mean big. We're not just talking about a little typo here. We're talking about gaining or losing an entire volume of that instruction manual.
So this is a really key difference in scale. You got to get this. Over on the left, we've got aneuploidy. Think of it like this. You've got one extra or one missing volume, like maybe an extra copy of volume 21. But then on the right, you have polyploidy, which is way more dramatic. That's like getting a whole extra copy of the entire encyclopedia. The scale of the change is just completely different.
And to make this aneuploidy thing really click, the most well-known example is trisomy 21. This is what causes Down syndrome. It's where a person has 3 copies of chromosome 21 instead of the usual 2. It's just a perfect, real-world example of how adding just one extra volume can have these huge effects on the final design.
But what about adding whole sets? I mean, that sounds pretty drastic, right? Well, while it's often lethal in animals, in the plant kingdom, polyploidy is a major driver of evolution. It's not an error at all, it's a feature. So many of the crops we depend on, think wheat, strawberries, they're polyploid. This isn't just some minor tweak, it's a full-on mechanism for creating entirely new species with totally new traits.
All right, let's zoom in a bit. So we're moving on from talking about whole volumes of the blueprint. Now we're going to get down to the nitty-gritty, editing the actual sentences and paragraphs within a single chapter.
First up, deletion. This is pretty straightforward. A chunk of the blueprint just goes missing. And this is a great way to think about the consequences. Just imagine a critical page is ripped out of an assembly manual. The thing you're building's probably going to have some serious issues, right? Well, in humans, the loss of just a tiny piece of chromosome 5 is what causes Cre-Du-Shaw syndrome.
Okay, next we have duplication. Here, a segment gets accidentally copied and pasted.
Think of it like a page getting stuck in a photocopier, and you get an extra copy. Now, this is a really profound concept. Yeah, it can cause changes like the bar eye trait you see in fruit flies, but duplication is also one of the main engines of evolution. Why? Because it creates a spare copy of a gene. That spare copy is then totally free to mutate and take on a completely new job without messing up the original. This is literally how entire gene families, like the ones that give us our sense of smell, are born. It's incredible.
All right, now for a real twist, literally. With an inversion, a segment of the chromosome breaks off, flips a full 180 degrees, and then sticks itself back on. So all the same information is still there, it's just backward.
And this analogy is perfect. Imagine one of the instructions in your blueprint was literally installed upside down. All the words are there, but the order is reversed, and that can really confuse the cellular machinery that's trying to read it.
And finally, for this section, we have translocation. This is where a piece of one chromosome breaks off and attaches to a completely different, non-matching chromosome. It's like it's moved to a new neighborhood.
So the crucial point here is that the information isn't lost, it's just in the totally wrong place. It's like taking a page from the engine manual and sticking it right in the middle of the electrical wiring guide. You can see how that would cause a lot of confusion for the cell. Okay, so far, a lot of what we've talked about can feel like, well, accidents or errors. But our genome also has built-in, programmed ways to change. This isn't about mistakes. This is about deliberately shuffling the deck to create variation. This is the engine room of normal, healthy diversity.
During the formation of sperm and eggs, our chromosomes do this incredible dance. You can see it laid out here. They line up and then they literally swap pieces. This process, called homologous recombination, is evolution's #1 tool for generating new combinations of traits. It's the reason a species can adapt to new diseases or a changing climate. It's not a bug. You could argue it's the most important feature we have for long-term survival.
Hey, if you thought swapping pieces was wild, get this. There are sections of our DNA that can literally pack up their bags and move to a new location.
These are the famous jumping genes, or transposable elements. They were discovered by Nobel laureate Barbara McClintock, and they make our genome an incredibly dynamic place.
And they basically have two main ways of making the jump, which this slide breaks down perfectly. You've got copy and paste, which makes a new copy and leaves the original behind, and then you've got cut and paste, where the original piece itself moves. So why does this matter? Well, because where these genes land can change everything. If one of these jumping genes lands next to another gene, it can act like a brand new switch, turning that gene on or off in new ways. And just like that, you've instantly created a potential new trait. So, after seeing all these ways our genetic blueprint can change, from what look like accidental deletions to totally programmed shuffling, it brings us to a really fundamental question. Are these alterations just glitches in the system, or are they a core feature of how evolution actually works?
And the answer, as this slide really brings home, is it's both. The line between what we call an error and an innovation is incredibly blurry. A deletion can cause Cri-du-Chat syndrome, but a duplication, another so-called error, provides the raw material for a brand new gene to evolve. The very same recombination that creates healthy diversity can sometimes go wrong. It's really two sides of the same coin. These aren't separate things. They are the messy, unpredictable, and powerful source code of all biological change.
So what's the big takeaway here? It's that our genetic code isn't some static, perfect manuscript carved in stone. Not at all. It's a dynamic, living document that's constantly being edited, rearranged, and revised, sometimes by accident and sometimes by design. And that leaves us with one last thing to think about. Are what we call errors just the unintended but necessary consequences of the very system that makes all of evolution possible in the first place?